By the early 20th century, scientists no longer wondered whether microbes existed. They wanted to know how they lived. Observation had evolved into measurement. The microscope revealed shape. Now, chemistry and mathematics would reveal life's rhythm. In laboratories from Paris to Chicago, glass flasks filled with cloudy broth became living clocks. Scientists watched bacteria multiply, die, and adapt, recording the rise and fall of cell numbers as growth curves. Each curve told a story in 4 acts: the lag phase of adjustment, the exponential surge of abundance, the stationary pause of exhaustion, and finally, the decline. Time and nutrients dictated destiny. For the first time, life itself could be plotted on graph paper. These curves became the grammar of microbial physiology. They showed that even the simplest cells obeyed laws of economy. Growth demanded energy, structure, and coordination. Temperature, pH, and oxygen each shifted the shape of the curve, revealing how microbes tuned themselves to their surroundings. The cell was no longer a mystery. It was a system that could be described, predicted, and eventually programmed. The search for what powered that system led to the discovery of metabolism. Using respirometers and calorimeters, researchers measured oxygen consumption, carbon release, and energy flow. They realized that microbes were chemical engineers, capable of running diverse metabolic programs. Fermentation, respiration, photosynthesis, nitrogen fixation. Some oxidized sulfur or iron instead of sugar, others survived on hydrogen gas. Microbial physiology revealed that life could extract energy from almost anything, that chemistry itself was alive. But microbes did more than grow. They changed. Mutations appeared spontaneously within cultures, creating variants that resisted heat, phages, or antibiotics. Salvador Luria and Max Delbruck demonstrated that mutation was not a response to stress, but a random act of chance, later selected by survival. Evolution, they showed, was written not in centuries, but in hours. The Petri dish became a theatre of adaptation. The next leap came from heredity. In the 1940s, Oswald Avery and colleagues proved that DNA, not protein, carried genetic information, using Streptococcus pneumonia as their model. Soon after, Joshua Lederberg discovered bacterial conjugation. Microbes could exchange genes directly, forming a primitive sexual network. Transformation, transduction, and plasmids revealed that bacteria were not isolated units, but participants in a genetic commons. The boundaries of individuality blurred. These findings reshaped biology. Bacteria became models for understanding all life's instructions. Through the study of E. coli, scientists uncovered the genetic code, the universal translation between DNA's language and proteins. The central dogma, DNA makes RNA, makes protein, grew from microbial logic. In the simplicity of a single cell lay the blueprint of evolution itself. By mid-century, growth curves had become digital, powered by spectrophotometers and automatic counters. The living clock turned mechanical, feeding data into equations that describe population dynamics. With every measurement, microbiology grew closer to physics, a science of laws and limits, not just forms. But within those equations remained the pulse of life. Cells dividing, mutating, adapting, endlessly rewriting their own survival. The story from growth curves to genetic codes is the story of biology becoming quantitative. It is the moment when life's patterns were not only seen but measured, modelled, and ultimately read. From a cloudy broth to the double helix, the path was one of clarity. Each discovery turning motion into meaning, each equation translating chaos into order, until the invisible became intelligible.